Good afternoon and welcome everybody. I'm Anu Gaba. I'm a medical oncologist and hematologist and the chair for cancer services here in Fargo for Sanford Health and for Roger Maris Cancer Center. I'm also a clinical researcher. It's my honor to have Dr. Carl June with us. He's the winner of the Sanford Lorraine Cross Award, which honors life-changing breakthroughs and innovations in medical science. And this is the award that was initiated by Sanford a few years ago. So it's really fitting that Dr. June is here for our inaugural All-Star Week, which really celebrates the vision for Roger Maris Cancer Center to be the national destination for cancer care, innovation, research, treatment, and training as we launch into our fellowship program within the next year. And the work that Dr. June has done is also very fitting with our goal of establishing this region's first and only bone marrow transplant program. So at this time, I welcome Dr. David Pierce, who is the president of Innovation Research and World Clinics at Sanford Health, to tell us all a little more about Dr. Carl June and his accomplishments. So thank you.
Thank you, Dr. Gaba. So, Dr. June personifies a lot of things in science and research. If we just take a step back, I proudly oversee the research for Sanford Health. And what does that actually mean? It means this: research. Every one of us, whether we're a physician, a researcher, a parent, a brother, a sister, has said these words: I wish more could have been done. 2 million people will be diagnosed with cancer in the next year in the United States, and that's where we clearly need to do more. And that's what research and individuals and pioneers like Dr. June do. So just to sort of level set in terms of the stature of this man, the Sanford Lorraine Cross Award it's not a way where we pick our favorite people. It's an algorithm that goes through the literature and all of the biomedical research findings of the last 10 years and then picks an area where there's been the most significant impact. Then a group of other scientists sort of help identify that individual. This is an award, and to use a cliche for today, for someone who's hit a home run, truly been a champion and a pioneer with respect to what they've done. So Dr. June received the second iteration of the Sanford Lorraine Cross Award this last year, slightly delayed because of why, we know why, in terms of gets-togethers because of our friend COVID. So what Dr. June now has done is, and he has his partner here, Dr. Bruce Levine, he will be presenting a little bit about the past, the present, and the future in terms of how chronic conditions such as cancers can be conquered. It's not just what he's done in terms of treating and curing individuals today, it's also about tomorrow. There are 300 companies, at least, that are developing the platforms on top of what his initial findings were. So I'm proud to introduce the 2020 Sanford Lorraine Cross winner, Dr. Carl June.
Well, thanks, David, and thanks, Dr. Gaba, and it's really great to be back with Sanford. And so we're going to tag team this. I'll give an initial overview, and then Bruce Levine, who I've worked with since the early 1990s, will then follow up with some other broader perspectives. And I don't know if these slides can be seen. It's really very bright here. And my conflicts are listed here. What I'd like to do is go over some types of T-cell therapies, which are this new kid in the block for cancer, and where they'll be applied here, where the next steps might be. And it all starts on what's on the right, which is the cancer wheel of how do you change how the body's immune system reacts to cancer. And it involves a complex series of steps, with the immune system responding. And I think public has learned a lot thanks to COVID. And RNA vaccines, it's done us a big help, I think, in that the lay public now understands a lot more about the need for an immune system, and really a lot of the issues in infectious disease are shared with cancer. But there are other issues that make cancer harder to deal with for the immune system than an infection. So you have to remember our immune system's evolved to fight infection, but it's only then more recent evolutionarily where they have also had a role in immunosurveillance and culling out early tumors. So there's now good data in humans and mice that occurs. But what we're concerned with here, is, people come in with late stage cancers, and how can you deal with them? Can you turn the immune system on something that it has been told not to attack? So there are three kinds of cell therapies in advanced development now. The first one was actually something that some of us trained in a long time ago, bone marrow transplants. So a bone marrow transplant is a highly personalized therapy, made of a patient's stem cells. But that's not FDA approved. It happened, in the practice of medicine, just like blood cell transfusions. When CAR T cells came along and other cell therapies, the FDA changed the rules and said, if you're making genetically modified cells, it needs to be FDA approved. And so the three types of cell therapy, the one used the first is called TIL therapy for tumor infiltrating lymphocytes. And that's really been done initially for melanoma, but it's basically where a patient has to have a metastatic cancer. The tumor gets taken out, and then the T cells that are in that tumor are grown up. Those are the infiltrating lymphocytes, the TILs, and then given back. So that's a much more complex procedure than the other two types shown in that cartoon, which is where you start with a blood and you just do like a blood donation, and it doesn't require surgery. But there will be, I'm pretty sure, FDA approved TIL cell therapies for things like melanoma and lung cancer, as well as CAR T cells and T cell receptor therapies, which start from peripheral blood. And, so TIL therapies started about 40 years ago, and, they're still not FDA approved. It just shows how long it takes things like that to happen. But they showed durable responses in patients who normally had a survival of 6 months, metastatic melanoma. And but everyone said it could not be commercialized. And about 2 months ago, I did a search on the web for TIL therapies, and there's about 6 or 8 companies now making TIL therapies. So you're going to first start here with CAR T cells, made by Novartis and Bristol Myers and so on. But there will be eventually many flavors. There'll be TIL therapies made probably by one of the companies on there. There'll be CAR T cell therapies and T cell receptor therapies, which are highly personalized. But the reason that they're being developed is they can actually cure patients who would not otherwise survive a cancer. So CAR T cells, that word CAR T cell comes from the abbreviation chimeric antigen receptor T cell. And that came after this, the Greek fantastical, creature that was a fusion of a lion's head, a goat's body, and a serpent's tail. So that was a chimera of 3 different species. What a CAR T cell is a chimera of a B cell and a T cell. And the B cell makes antibodies, and normally a T cell doesn't. And T cells are really good at killing cancer cells. So a fusion of those is what a CAR T cell is. So it doesn't exist in nature. It's made by taking your own cells out and then inserting the antibody genes that the CAR B cell would normally make. And then you have this chimeric cell. So that's where the term comes from. Bruce Levine and I did the first CAR T cell trials actually for HIV. And this was back in the early 1990s because there were no drugs back then. And people died like flies from HIV because their immune system was basically destroyed by the virus. And it turned out we showed how you could grow T cells in patients with HIV. And then, ethically, the first place to test it wasn't cancer because there had never been T cell transfusions like this before. So we started in AIDS patients who had no T cells. And we showed there was a dose-dependent response in the patients. So the more T cells we gave them, the more their immune systems got better. And then so that was called a first generation CAR because of molecular designs of the CAR molecule. We got better and better in the field. But those first generation CAR cells actually had effects in patients. And when Bruce and I we were initially in the Navy and moved to the University of Pennsylvania in 2001. But in the 90s, when the AIDS epidemic was at its peak before the drugs came out, we did these initial trials in the Navy, and with the team shown here. And the FDA has had a policy that if used gene therapies, and David Price is going to do those for some congenital diseases. Unlike other therapies that the FDA develops, the FDA has a policy that you need to follow the patients for 15 years. So there's never been prospective databases like that before, but we had that in the first CAR T-cell trials. And, where our AIDS patients came back from their first infusion in 97. And then we started, we had this, freezer bank full of cells. So we thought the CAR T-cells were gone in all the patients because we'd just given them a single infusion in 1997. So we went to the freezer and analyzed those samples and found that those CAR T-cells were alive and well in the patients a decade after we'd infused them. So a single infusion led to at least, by mathematical modeling, it was a 17-year half-life. So we don't have that kind of follow-up yet in our CAR T-cell cancer patients. But at least in AIDS patients, we know two things, that it's a so-called living drug. A single infusion of these can last at least a decade. So that's very different than all the pharma is, where you give T-cells or any drug and it goes away, it's metabolized, and you have to give it recursively. So engineered T-cells are like a stem cell, and they can last, we think, probably decades in patients. And that then is one thing and reason why cancers don't come back, because it's not you give the therapy, but it acts in a way like a cellular vaccine and prevents the cancer from coming back. The second thing we learned is there were no long-term toxicities in these patients. We had 40 patients on the trials, and none of them had problems from T-cells gone haywire. So we think that data applies to the cancer patients because we're using the patient's own cells, called autologous cells, okay? That could be very different if you use someone else's T-cells and park them in someone else. But the safety of autologous T-cells is very safe. And we know that from long-term bone marrow transplantation data where donor lymphocyte infusions are given. And by extension, autologous CAR T-cells probably will have long-term safety effects, but we need pharmacovigilance to really establish that. What this slide shows is the difference between a CAR T-cell and a T-cell receptor T-cell. Both of them are, going to be FDA approved. The T-cell receptor T-cells are being worked on by GSK, and you'll see FDA approval probably for ovarian cancer. They're more complex. They require HLA matching. And then the T-cell receptor is what's put in with the gene transfer technology. With CAR T-cells, you don't need to know the patient's HLA type. It's just a stand for the same CAR molecule that works in one person works in everyone. And that's the beauty of an antibody and why B cells are a good thing to fuse with T cells. But we're going to see both because I think they're going to be complementary. And let's see, maybe I better do. So, this is a slide basically showing how, in the history of medicine, how long things can take. And Cicero said, in 100 years BC, that if you knew the cause of some disease, then the treatment would follow. And in Philadelphia, we had back in 1960 the discovery by Peter Knoll of the first genetic cause of cancer. He found that what's called the Philadelphia chromosome, and that's a fusion of two genes that lead to an oncogene. And then later the treatment with imatinib. And the lesson there is he discovered that in about 1961, and it took almost 40 years to get a treatment for it. So with the exception of the COVID vaccines, where the trials went very fast, it takes a long time, even when you know the cause of a disease, to get an FDA-approved therapy. And with cell and gene therapies, it's not been an exception. This is the timeline of the approval of Kymriah, which is made by Novartis, that Bruce and I made and tested first at Penn, and then Yescarta, which is now marketed by Kite Gilead. And basically, our first patient was reported in the literature in 2011. We treated them in 2010. And then it led through a whole series of events on this slide to FDA approval in 2017. That's actually fast by pharmaceutical standards, and much fast. So that's a good thing. What I can say is, now immuno-oncology is a huge $30 billion industry when you give the checkpoint therapies, which cure 20 or 30% of people, but there's 70% that aren't cured. We're about 10 years behind that with the cell therapies, because the first checkpoint therapy, FDA approved, was ipilimumab, anti-CTLA-4, and then PD-1. That started in 2011. So we're about a decade behind in that, and I think, if you map 10 years down the line, you'll see the same kind of growth in the cell therapy industries that you're undertaking here is what we've seen with with checkpoint therapies. So this is, where we began back in 2010. Our first patient was infused in July 31st on a protocol with chronic leukemia. And then we reported in 2011 in the New England Journal of Medicine. All of these are, showing this drug production cycle where the patients would give blood donation, usually in our apheresis center. And then 10 to 14 days later, the cells were manufactured at the University of Pennsylvania. Now it's at commercial centers. And then, the patient is at the infusion room, and we treat patients inpatient or outpatient, depending on their clinical condition and what the CAR molecule is. So but in all cases, basically, we just have a 37-degree water bath in the room. The cells are thawed for about 5 minutes until the last ice crystal's gone, and then they're infused in a 5-minute infusion. And you're going to be seeing that a lot, I think, in a year from now, from what I've heard. And I think it'll be exciting. It's actually anticlimactic. The infusion is a zero non-event. And then the patient is, if they're healthy, they go home, and then you wait, and do they have cytokine release syndrome or not, which happens, in the following week. But the infusions are very well tolerated and not a clinical event. So led to FDA approval in 2017. And then this just shows the commercial landscape right now. There are, right now in the US, it's either Kite Gilead, Novartis, or now Bristol Myers making CAR cells. They're all autologous. They have some design differences with them, and then the different indications that are listed here from lymphomas that are refractory and relapsed to various kinds of acute leukemia. And there's many on the horizon that will happen on their labels. And this just shows the trials that were done. All the FDA approvals came off non-randomized, multi-center phase 2 trials. And they got FDA approval because they were done on patients who had failed all, FDA approved therapies. And so in hematologic malignancies, the FDA standard is 50 to 100 patients is usually enough. It's going to be different when they go to more upstage or, for frontline therapies. But those are the trials that led to approval. And what we're finding out across the board is about half the patients look like they're cured. And there are a number of reasons why half are not cured. And some of them, and it depends actually on the disease. The resistance mechanisms in lymphoma differ than from non-Hodgkin's lymphoma. And there's many next-generation CAR T cells that overcome this. We have a 90% complete remission rate at the University of Pennsylvania for leukemia. Just 2 weeks ago, this came out on an announcement by Bristol Myers timed with the ASCO meeting. It's the first randomized controlled trial with CAR T cells, and they did it in patients with large B-cell lymphoma that was refractory, in second-line therapy. And what they did was compare it to autologous bone marrow transplants, which are right now the standard of care for that disease. And they found out that it met all the primary endpoints. CAR T cells were better than autologous transplant. Now that at this point is a press release. It hasn't been peer reviewed, but I think likely that was a international multi-center phase 3 trial. So it's likely that the CAR T cells are going to move to near front line here and there's about at least 60,000 patients a year in the United States that get refractory lymphomas. And so you will see that'll be the dominant population of adults. And there are trials to move it to front line rather than second line here. But so many trials like that underway. Also during COVID, the other major watershed event was the approval of, and by Bristol Myers, of a CAR T cell for myeloma. And myeloma is, the most common malignancy in adults, and it's a horrible disease for the hem-onc people who've seen that. All the textbooks say it's non-curable. There's a very high initial response rate. The CARs in myeloma target a protein called B-cell maturation antigen, or BCMA. All the previous CARs I mentioned for cancer target CD19. So now we have 2 kids on the block, if you will, BCMA directed, which is primarily for myeloma, and then CD19, which is for acute leukemias and lymphomas. And then the problem right now with myeloma is that most of the patients still relapse after 2 or 3 years. They don't seem to see the plateau on the curve like they're cured. So there's a lot of room to improve in myeloma, but right now, it's much better than what they used to have. And so this just shows then those clinical trials. So this is to see if you're paying attention, a learning check. What is the typical time to response for patients who have a CAR T cell therapy? The answer on this test would be 1 to 2 months. And the reason for that is that's when imaging is done, or you do a bone marrow. Okay, so if you have acute leukemia, a month later, you get a bone marrow. If you have lymphoma, a month and 2 months later, you get imaging. The answer in reality is days. So you can watch, I've heard Stan Riddell mention, patients with lymphomas, he says it's like ice melting. So you can see lymph node masses, which just go away within days after treatment. So the responses are, in our leukemia patients, 2 weeks, and the patient's leukemia is gone if you do a non-standard clinical assessment. So the responses are much more rapid than they are with chemotherapy, but by imaging, we don't pick it up, and we don't know until you do that. But there's another thing that will change, which is monitoring the CAR T cells in the blood, and this is a whole new area of pharmacology. If the CAR T cells are proliferating, and if they have some kind of fever and so on, that's a cytokine storm, most likely those are the responding patients. The patients who get no fever, or if you look in the blood, they don't have CAR T cells, they're the ones who aren't responding. So we're going to have all kinds of companion diagnostic tests that we don't yet have. And it's really important to have those. And so patients who respond to CD19 CARs get an acquired B-cell aplasia, so their normal B cells go away, as well as their malignant B cells. And that's called an on-target but off-tumor effect. And so by using flow cytometry, you can find that out. If their B cells don't go away, the CAR T cells did not work. Okay, so it's very different than assessing the effects of a chemotherapy drug. We have here, real thing. And same thing if you look in myeloma, if their BCMA positive cells didn't go away, it failed. And you need to go back to the drawing board. So the responses are very rapid clinically, but then you assess it a month or two later. And with the Novartis drug, it was the first time they ever approved a very expensive drug. It's about $400,000 per patient, but you only pay for it if it works. Which means when you do a bone marrow assessment later, if they didn't get a complete response, you're not responsible for that product. So that never happens with chemotherapy, where you get performance, you pay for performance. But that's a big issue, is the cost of these therapies. So the first patient we ever treated in 2010 is shown here on the upper right. He had refractory p53 deficient CLL, and he came back in the midst of the pandemic in August last summer, still in remission. And we haven't published this yet, but we're getting ready to. He still had CAR T cells 10 years later, and he was still in remission. So we think, his CLL was cured. So that's, and the same thing with a second patient. He came back last summer, and he is in remission as well. So, and then we began in 2012 treating pediatric and young adult leukemia, which is more rapidly progressing than CLL. We had to start by FDA mandates in adult leukemia, and then we went to pediatrics up to age 25, and published that in 2013. And that led then Novartis to do this international single-arm trial of relapsed refractory pediatric and young adult ALL, called the Eliana trial, and that showed an 82% complete remission rate. And then FDA approval. And then this is the actual initial 60 patients we treated in Philadelphia at Children's Hospital, with the swimmer's plot showing the green lines are complete remissions. The oldest farther out on this graph, which is not updated, was to 48 months, and that was Emily Whitehead. But almost all 90% had a complete remission. The only cases of relapse really were if the tumor cell lost the target, CD19. And that's leading to a whole kind of new therapy, which is basically using a cocktail of CARs, targeting molecules, so that if one is lost, the leukemia can't escape by losing its target, which might be, in this case, CD19. So that was unheard of, to have a phase 1 trial have a 90% complete remission rate, and that's why Novartis decided to do the international trial. Now we have something even better, and this isn't published, but on the left is what Kymriah is. It's an antibody that binds CD19 made of a mouse antibody. And then it has the signaling domains. It's called CTL019. On the right is something we call CTL119. We made this with Novartis, and it's fully human. It doesn't have the mouse antibody. And and this data is not published, but pediatric and young adult in patients who've never been treated with a CAR T cell, we got 100% complete remission rate, 42 out of 42. And this is something I brought up. What motivation does Novartis have now to take this to FDA approval? We think it's better. Okay, it has basically 100% complete remission rate, but it would displace what they spent a lot of money getting FDA approved already, in 2017. So this is a big frustration to us. And the other bane, we've always talked about toxicity with with chemotherapy. The major issues being off-target toxicities like, alopecia, vomiting, and all that. With CAR T cells, the issues are on-target toxicity, which the patients accept. That's a cytokine fever and so on that we know how to treat now. But there's a new kind of their problem, which is this financial toxicity. So the average cost of an FDA-approved drug for cancer in the last couple of years for any drug is $150,000 a year. And it's not sustainable. So it used to be that drugs were only 20% of our medical bills across the US, and 80% was hospital visits and lab follow-ups and all that. So the price of drugs is going up. And what that really says, I think, is we need one-time treatments, rather than a bunch of things that fail. And then that'll lower the total cost we do. So so this is this emerging issue of financial toxicity and how will we afford it? And our system has to figure that out with improved manufacturing as well as not using therapies that don't work. So this was made by Shannon Mott, our colleague at Children's Hospital, and she lists the toxicities of CAR cells for acute leukemia. And there are not that many. There's the acquired B-cell deficiency, so-called B-cell aplasia, and transient increase in infections and so on. And then the fact that I mentioned earlier, we don't know long-term side effects, but right now we do know from HIV that autologous CAR T cells have no long-term complications. The complications of standard chemotherapy for acute leukemia are awful. It's a long list that goes all the way down this, and it's actually worse in pediatric patients than adults. So radiation and so on, is worse on children who are trying to grow than it is adults and so on. So hopefully we'll be able to replace these kinds of therapies that are very toxic with long-term side effects with immune-based therapies, which should have less long-term toxicity. So I wrote a review last year in the New England Journal about this new issue of cytokine release syndrome, also called cytokine storm. And it affects nearly every organ, and it's a clinical diagnostic challenge for the clinicians and who need to figure out, is this infection, is it cytokine storm or not. That's a very important skill to learn, just like diagnosing graft versus host disease is. And there's a number of cytokines and so on that are perturbed and shown on the right. But the initial therapy is tocilizumab, which blocks IL-6. And that got co-labeled by the FDA to treat emerging cytokine storms. And then it's fully reversible. The package label for Kymriah is shown here, and it has black box warning for two things: the cytokine storm, which you have to learn how to manage, and right now is quite easy for experienced hem-onc physicians to diagnose. And then the second was a surprise, which is reversible neurotoxicity. It can present with aphasia, confusion, disorientation. And there's another one which is listed as number 7 there, which is the Kymriah is based on an HIV-based lentiviral backbone. And so the patient can have a false positive test for HIV because the genetic sequences for the CAR molecule have some of the residual ones from the wild type virus. So there's no AIDS virus in the patient, but some test will give a false positive, and we tell the patients ahead of time about that. So this then is from the transplant groups on how you grade cytokine release syndrome. There's no true ways within the toxicity tables that actually, because it's really a pretty new syndrome in medicine, this cytokine storm. It never really happened much like it does in CAR T cell therapy. And by the way, it happens in both CD19-directed CARs as well as BCMA-directed for myeloma, and we've seen it in solid tissue CAR trials, for instance, in pancreatic cancer and prostate cancer. So there's a grading system for this, and you need to know when to admit a patient. We have several ways to follow the patient that I heard you were putting in place here. There's also the CNS grading for this. It's called ICANS. That side effects And, you look at things like orientation, writing ability, following commands, and scale, the icons they have. And then that's treated in some ways different than cytokine storm is. And this is the grading scale then put together for adults. And again, we've seen ICANS both in CD19, directed CAR therapies, and BCMA. So some of the mechanisms are common depending on, even though you're targeting two different antigens, BCMA or CD19. And then this is just a cartoon showing the possible and theoretic side effects with CAR T cell therapy. And I talked about the cytokine storm. There are, thousands of patients who've now been treated, and no one has had where the T cells themselves transformed. So one theoretic risk is insertion. It's called insertional oncogenesis, where when the lentiviral vector or the gamma retrovirus puts the CAR DNA code into the patient's T cells, that could make the T cell be transformed into a T cell lymphoma, or a leukemia. That's never happened. It will probably happen eventually. It has happened in experimental trials with stem cells. The French had where they were treating some severe congenital immunodeficiencies, so-called SCID kids. They've had cases where the gene-modified stem cells became leukemia. It appears that the risk of that is cell type specific. So it's never been seen when mature T cells are genetically modified, but it has been seen with stem cells. So anyway, that's just the state of that. The neurologic toxicity has been really puzzling why it occurs. This is just some data we found last year in a paper in Cell that the molecule CD19, if you look up in all the textbooks, say that it's only in B cells or in leukemia. And what we found was it's in a molecule that goes around the blood vascular. They're called pericytes, not parasites like a malaria, but a pericyte is a cell that lines a blood vessel. And only the ones in your brain express CD19. The blood vessels in your arms and lungs don't express CD19. We have no idea why that's true, but we think that's one contributing mechanism to ICANS, is that the CAR T cells can help get into the brain by going through the blood-brain vasculature. So coming back to baseball, Yogi Berra said, you can learn a lot by watching. And that's how we learned about this ICANS syndrome, CD19, by studying the patients. And he said, if you come to a fork in the road, take it. So he had a lot of wisdom. And, our story has had a lot of forks in the road. The new ones will be using CAR cells in non-T cells. So right now, it's all peripheral blood T cells because they're the ones that evolved in your bodies to fight infections and can kill cancer cells, but there's natural killer cells called CAR-NK. There's CAR in macrophages, and a macrophage doesn't kill a cell the way a T cell does. A T cell punches a hole in the tumor, and then it dies. Macrophages actually eat, in the process, phagocytosis. So they, in principle, should be pretty synergistic with a CAR T cell, and we're testing that. We're testing expressing CAR molecules in stem cells, and various groups and biotechs are testing CAR T cells and induced pluripotent stem cells, where you could grow up one big batch and then give them to many different patients. And then CAR T cells are not just going to be used for cancer. They're being used, and there's references here for HIV/AIDS, as I mentioned, and then there's ongoing studies. They're being used for autoimmunity, and organ transplants to prevent organ rejection. And we had a paper last year in Nature showing that CAR T cells could prevent heart failure in mice and promote regeneration of injured myocardium. And so I think there's many different, applications of these of engineered cells like that. So, okay, you're going to be certified as a place for administering CAR T cells. And these slides will be online I'm teaching a course for the Society of Immunotherapy of Cancer. This shows places, and you will have to have this REMS, which is risk evaluation and mitigation strategy. You got to follow the patients. And, each of the products, so it's different from the Kymriah product. We give that as an outpatient. We admit the patients from Gilead. They have more rapid and more severe cytokine storms. So how you manage the patient depends on which product they get. And then you need to know that you know how to manage the toxicity. So this is the REMS requirements for giving CAR cells. And you need to have 2 doses of tocilizumab on site in your pharmacy. That can be a huge issue if you don't have it. And this just shows down the line what you need to know about, and then following the patients following as part of the pharmacovigilance. So there's a lot of training that's involved, and you guys are going down that road now. We require that the patients have a 24/7 caregiver for 8 weeks. And we give them this wallet card that you can see on the right-hand side, so that if they get a fever, for instance, they could go to some primary care doc who doesn't know about CAR T cells and get entirely the wrong therapy for what they have. So it's really important that the patient get educated as well. And, it requires a whole team, which is what you're putting together here. And this just shows from A to Z, from apheresis, cell therapy, clinical care, ICU, the financial services, et cetera. All this has to be in place to be efficient at this. So, which is an incorrect guidance for after CAR T-cell treatment? You need to have a 24/7 caregiver. They have to be within 2 hours of the facility, and I learned today you'll have a local hotel, because if they get a fever, they can't go a long time without treatment. And they have to have a wallet card. They don't have to, you don't have to tell them they can't operate farm machinery for 6 months. We have had one of our patients become an Olympic level. He was a hockey player in college, and he went back playing a month later with leukemia. So if they respond, they return to normal health in a month, okay, unless they have other issues. So it's not like a bone marrow transplant where there may be graft-versus-host disease issues and so on. So they can go back rapidly. There's no long-term immunodeficiency. So this is where, Bruce and I wrote this review a few years ago in Nature about the healthcare system going from 3 pillars, which is the pharmaceutical industry, the biotechnology industry, and devices like hemodialysis, to erecting the 4th pillar, which is cell therapies, which you're in the midst of doing. And there's a lot of issues. Right now we use the patient-specific so-called N-of-1 manufacturing, and sometimes this will be done in blood banks. And I think in the future, locally, you'll be doing manufacturing. Right now, it's done in a pharmaceutical manufacturing plant, and it requires it shipped back and forth. But all that's going to evolve over the next years. And then CAR T cells are becoming global. When we started, they were only in the U.S. at 3 centers, and now there's hundreds and hundreds of trials underway testing CAR T cells. And one thing that will really disrupt the industry is if we can make so-called universal CAR cells, and other people call them off the shelf. These are being tested both in academia, including where we're at in Philadelphia, and biotech. And that could be a huge thing. If you have cells pre-made in the pharmacy and an acute leukemic patient comes in, you can give him cells that night, and you don't have to wait 2 or 3 weeks to manufacture the cells. So that's on the horizon, and I think we're going to have both cells like that, the autologous, patient-specific, as well as off-the-shelf cells. So we're at this time, where the paradigm's changing in medicine, from in the space of oncology, from chemo, radiation, and surgery to precision-based medicine. And in the long run, it should be both more efficacious and less toxic. That's the revolution we're in. And so that's a summary of what I've gone over. A lot of people, both at the University of Pennsylvania, Children's Hospital, were responsible for the trials I showed, and our collaborators with Novartis, Stand Up to Cancer, and the Parker Institute. And so I'm going to turn this over then now to Bruce, who's going to give you his perspective, both of manufacturing over the last 30 years, and then he's president of the International Society of Cell Therapy now for 2 years. Thank you.
Okay, can you see slides? Great. So I can't tell you how wonderful it feels to give a talk to real live people in 3 dimensions. I haven't done this in quite a while, so I have just a few thoughts to share with you, some things you should know and some things you should think about. Let's see. Clicker. Why is my presentation not moving? Let me try and unplug and replug. Yeah, I may have a frozen
So
PowerPoint.
it's looking just like that.
Yeah.
Yeah, that's what this does.
Yeah.
Maybe, did you unplug it and then it still did that?
Yeah, I did.
Yeah, why don't you unplug it and see if it, might go back to.
There we go. Okay, so I have a financial interest due to intellectual property and patents that's managed by university policy. So this is a very good book about precision engineering that I recommend to you. And it talks about the evolution of technologies from steam engine pistons, cameras, space telescope, GPS, photolithography. So over time, we're getting better and better. CAR T cells are not this. They are not precision engineering. So you're going to have to learn to live with variability and some certain unpredictability. So how do we make CAR T cells? Well, it starts with the patient and collection by leukapheresis, volume of blood that may be processed 7 to 10 liters, sometimes 10. That bag goes to our manufacturing facility at Penn for commercial cars. It will go to Novartis or to Gilead or to BMS or to whoever has the next approval. And what they're going to do is to separate out cells that they don't need and take the T cells and deliver the gene encoding the chimeric antigen receptor using the lentiviral vector. That's permanent modification. Grow those cells up in a bioreactor several days, remove samples for quality control testing to address FDA criteria for safety, purity, potency, and identity. Once those tests are performed, then the cells can be released. In that time that the testing is performed, those cells are cryopreserved. So they can actually be sent anywhere in the world. And that's how Novartis manufactures cells in New Jersey for patients in Australia and Japan and Europe. But we know that non-T cell contaminants negatively impact making a product. And I've listed the contaminants that we see and some references here. And why? Well, peripheral blood counts differ by clinical indications. So you can see why blood cell count, platelets, lymphocytes, and monocytes. Look what happens in CLL, but that's not a good thing because all of those are B cells and you need to separate out the T cells. This is what we think about in manufacturing. What do you need to think about in the patients? Well, T cell fitness declines with more treatment cycles. The gray parts of those bars are the terminal effector memory cells that don't grow very well, that don't function very well. What you want are the green and the red and a little bit of the blue. So every cycle of treatment a patient gets, you get cells that are less able to generate a product and perhaps induce a complete remission. And not all collections and manufacturing runs are predictable. Here's another slide showing you some of the variability that we get. Look at the T cell percentage, the lymphocyte percentage, the monocytes, and it's all over the map. Now the blue are successful manufacturing runs. The red are where we were unable to generate a product. And while you see that there's a statistical difference, there's still a high degree of overlap. So how can you set a bar to exclude patients that might benefit? It's very tricky to do. So would you disqualify the patients in the green boxes from manufacturing if it turns out that you can generate a product? And here's a growth curve just showing sometimes you get cells from patients that just don't grow. They won't expand. What about timing? Now, I mentioned the subsequent rounds of treatment, and here's a patient that came in. He had an ALC of 300, which is just about on the border, maybe a little low, on December 23rd when he signed his consent. When his apheresis occurred, January 18th, his ALC was 100. And we didn't get enough cells to generate a product. So what happened? Well, he received some intervening treatment, including ibrutinib, cyclophosphamide, melphalan, and then a second T-cell collection at the end of April. And on that date, his ALC was 900, and we were able to successfully generate a product. So these are some of the communications that you're going to be having with the teams that are going to be working with you on CAR T-cell commercial and clinical trials. What about the final product? What do we think about characterizing the final T-cell product? Does dose matter? You dose imatinib, you dose ibrutinib, you dose blinatumomab. What about CAR T-cell dose? Well, here's our first clinical trial in chronic lymphoid leukemia. There is no statistical relationship between the dose of CAR T-cells that those patients got and their clinical response. So how do we think about dosing a drug that actually divides? But it turns out in pharmacokinetic analysis in vivo that there is a relationship between exposure and expansion of the CAR T cells and the response. And this is work that we did with Novartis, doing the correlations in pediatric ALL, in adult ALL, and in CLL. What you see on the left is area under the curve for the first 28 days, and what you see on the right is the Cmax, or the max concentration. There's a statistical correlation in each disease with clinical response. But still, there are some patients that fall outside the standard deviation. There are outliers, and what this tells you is that there are a multiple of effects, both T-cell intrinsic effects, product effects, tumor effects, and others such as trafficking. Viability. We have a viability cut point of 70% for our clinical trials. The commercial Novartis product is 80%. There was some kerfluffle on the ability of Novartis to manufacture products. It had to do with the viability set point. So then we looked, if you have a viability 80 and over, or 70 or 80 or below 70, does that correlate with clinical response? No, because it's the number of cells that are dividing that gives you that area under the curve. So thinking about wider patient access, and Carl and I have a commitment not just to innovation, but to dissemination and providing wider patient access. And that's why we're so pleased to be here today to work with you to open your center. We do need to work on enhancing potency, especially for solid tumors. We need to integrate so-called armor for T cells, switches, and combinations, combination CARs, and combinations with other therapies. These are complex drugs to manufacture. There may be a delay in the time that you send out a product to the time that you get it back. These are cell-based living drugs. We need to implement automation and shorten ex vivo culture. We actually have opened up from our standard 9 or 10-day process. We now have a 3-day process in a clinical trial that is open and treating patients. On a per cell basis, those cells are more potent. We need education and training at all levels. Carl mentioned I'm president of the International Society for Cell and Gene Therapy. The ISCT, I'll just tell you briefly, is a unique translational society that's global and has 3 pillars in the translational space. Scientific committees focus on multitude of cell types. We have regulatory and quality operations, and we have commercialization. And that's the thing about these advanced therapies. You have to have knowledge in each. You can't have knowledge exclusively in the science without understanding the regulatory and the quality aspects and the implications of commercialization. So we just had our virtual annual meeting. It was supposed to be in New Orleans, but it was wherever you were. We have a very nice interface, and it's available until December 31st, 2021. So if you go to ict2021.com, you can see all these lectures about regulatory quality operations as well as the science. We had a presidential plenary where we had Peter Marks, the director of the FDA CBER, Ari Beldegrun, Matt Porteous. I also want to tell you about a organization that was jointly founded by ICT and a sister society, ASTCT, and that is the Foundation for the Accreditation of Cellular Therapy. And this is focusing on the quality aspects. In FDA terms, you have to say what you do what you say, prove it, and improve it. And it's a lot harder than it sounds. And just reviewing some of the benefits of FACT accreditation, it's voluntary, but it's almost certain that the commercial car companies are going to require FACT accreditation. And health insurance plans and government agencies take this into account for reimbursement. I'll just list some of the others there, including the U.S. News and World Report takes this into account in their ratings for cancer care. These are the parent organizations, ICT and ASBMT. There are laboratory standards, there are clinical program standards, there are collection standards, and processing standards. I won't spend more time on FACT, but happy to talk to you more about it offline. Okay, so now we talked about education and training. Carl mentioned these are financially complex therapies. They're expensive upfront for long-term durable benefit. Novartis attempted and is attempting value-based payment that links price to outcome. I think with the increase that we're seeing in predictive biomarkers, we're going to see implementation of some of that to select patients that go for CAR T-cell therapy versus other therapies. So I want to close just with two heroes and two stories. You know the story of Emily. She was our first CAR T kid, actually the first CAR T kid in the world. 9 years later, she's cancer-free and an honor student with straight A's. Sophia is on the left. Sophia was enrolled in a clinical trial, and she had a complete clinical response. But then she relapsed with CD19 negative disease and unfortunately passed away from that. And her mother, Amy, just 3 months after Sophia's passing, drove from Kentucky to Maryland to testify to the FDA committee to recommend that the committee advise the FDA to approve Kymriah as the first CAR T-cell. So the lesson is that we need to learn from all patients, and we need to work on innovation and improving where we are to benefit as many patients as possible. So these are acknowledgments. It truly does take a village. This is a picture of the flash mob that was in the Abramson Cancer Center on the day of FDA approval, August 30th, 2017. You will not see me in this picture because I was in Boston that day giving a talk on CAR T cells, so I had to enjoy it vicariously. Usually we say we like the clouds to go away, but it's actually pleasant to have the clouds come in, give a little bit of relief. So with that, I'll close. Thank you for your attention, and if there's time, I don't know if we're taking questions or discussion.
Absolutely. So that was some great insight for past, present, and future, like I introduced the speakers with respect. I think the accreditation piece is something that's really something that this organization has taken seriously and done very well. So with that, do we have any questions from the audience? We have some time for questions for Dr. June or for Dr. Levine. There's a microphone coming your way, Dr. Gaba. Okay.
So how much of, what is the acute mortality of CAR T-cell therapy, I mean, within the first 6 months, not from their disease, but from the treatment itself?
Dr. Gaba, that's a great question. So it depends on the experience of the centers. Unfortunately, there's anecdotal, where, like I know a case of one of the first patients, myeloma in China. So we infuse the patients usually on a Monday or Tuesday, and then the cytokine release syndrome will usually be manifest by Friday. Well, they did it on a different day in China, and the patient had bad cytokine release syndrome on a weekend. They didn't have tocilizumab, and the patient was treated with steroids and antibiotics instead of cytokine, anti-cytokine directed therapy, and the patient died. So their first patient died because of inexperience. Now, Bruce mentioned we are working on implementing CAR T cells in low and middle income countries. And I've gone down to Costa Rica, and we're going to be infusing CAR T cells in Costa Rica. And just recently, and Bruce and I are working with a small biotech called Immunil in Bangalore. And the first CAR T cell, there's 1.3 billion people in India. The first person was just infused with a CAR T cell there. And so there's a learning curve, but that's not new to medicine, as you know. I mean, graft-versus-host disease with experience, it went from a lethality that was 20-30% to now it's under 2%, for an HLA match. And so our only mortalities with CAR T cell therapies in hematologic malignancies are really due to underlying disease or infection. So if the patient comes in with a previous fungal infection, that can reactivate, that's similar to bone marrow transplant. So There's very rare cases, it's under 1%. We've just published, it's unfortunately harder in acute leukemia, where the patients usually are sicker than others. So it depends their condition going in, performance status, and that requires some experience to pick the right patients. But in almost all the studies, the mortality rate's less than 2%, and that's then training dependent and age dependent. So, older people with acute leukemia have a higher, toxicity and mortality than younger people do. But that's not different from any other kind of therapy.
Thank you.
So for those of you streaming in by video, it started to rain here, just so you know. If the picture's not quite the same quality, there's a big plastic bag being wrapped around the camera right now. So, yeah, next question.
How critical the volume of the disease with the quality of response and the duration of response? When the patient comes with the high volume disease versus low, medium.
So, that's a really good question, which is there a relationship of response or toxicity based on the burden of disease? And both for the Gilead Kite drug and for the Novartis Kymriah in leukemia, there's a very definite relationship to the amount of blast infiltration in the bone marrow to the severity and incidence of cytokine storm. And we have found no difference, though, in the response rate for acute leukemia. There's probably a difference in some of the lymphomas, because of differential trafficking to nodal masses and so on, or extranodal. And in some studies, higher disease burden is lower response rate, but not all. So toxicity is related to, very close, as Bruce mentioned, to the proliferation of the CAR cells. And usually at the peak proliferation, the so-called Cmax, that's when you see the clinical manifestations the most. And so our higher, we're also seeing this thing of stage migration. Our initial patients literally had 5 to 10 pounds of tumor, and very late stage patients, and there was a 30% or 40% rate of grade 3 or 4 cytokine release syndrome, and now it's well under 20% because patients are being referred earlier with lower disease burden. So that's where we're at now, and then there's better care now and the ability to get cytokine measurements so that you can do preemptive therapy rather than reactive therapy.
Okay, so thank you. With that, with the other festivities today and with the weather, it's time to close out today. So Dr. Gaba, did you have any closing remarks that you would like to finish with? Thank you so much for being here, gentlemen.
So a heartfelt thank you to Dr. Carl June and to Dr. Bruce Levine for being here today. And thank you all for coming today. And we really welcome your participation and your questions. Thank you.